speak about a very important concept, that is restriction enzymes. You may get some questions from this concept. Okay, listen to me carefully. Restriction enzymes are nothing but a type of enzymes which normally cut the DNA. But still there are certain confusion. First we will understand the concept. Okay. The first let me just write who discovered this restriction enzymes? The scientist. Arber, actually let me write his full name, hope it is visible to you. Werner Arber discovered the restriction enzymes. Okay, he discovered restriction enzymes for the first time. Note this point. Okay, next is fine. We will erase this. I need space. Note. Werner Arber discovered restriction enzymes. What is exactly the restriction enzymes are called? They are called as molecular scissors. Okay, they are called as molecular scissors. Even molecular knives. Why? They have the ability to cut the DNA. But where exactly they can cut the DNA? That's what we'll understand. Okay, and all these restriction enzymes belong to a large class of enzyme family called as nucleases. Note this point. They belong to a large class of enzyme family, that is nucleases. Important, very important, nucleases. Okay, when exactly these restriction enzymes were discovered by this Werner Arber? Let me just write. In 1963, when Werner Arber actually he was conducting some experiments on E. coli. Let me just write E. coli. He was surprised when he treated E. coli with certain, okay, we'll draw it. E. coli, this is E. coli bacteria, with certain bacteriophages. Hope you know the meaning of bacteriophages, that they are nothing but the virus. Phage bacteria. That is, they are going to attack the bacteria and kill the bacteria. Okay, so bacteriophage, because they can insert their genetic material and use E. coli or any host organism as their factory, so that they can synthesize more proteins. Actually, this is it. Okay, the bacteriophage. And before 1963, everybody knew that when any bacteria, if it is treated with the bacteriophage, the bacteria will be killed. Okay, that is actually the assumption. Like it was proved also. Like the bacteria will be killed by this bacteriophage because of the lytic phase. Lytic means they will burst the bacteria. Got it? Once the number increases inside the bacteria. This was known earlier, before 1963. But for the surprise of this Werner Arber, when he treated this particular E. coli with certain bacteriophage, the E. coli showed resistance. That means this particular bacteriophage couldn't insert its genetic material. It couldn't insert its genetic material into E. coli. That means it failed. The bacteriophage failed. Then actually, when he studied the E. coli, he came to know that inside the bacteria there were certain enzymes. Okay, if the bacteriophage could insert its genetic material, these enzymes will break this genetic material into the fragments. Genetic material of the bacteriophage, I am talking about virus. Okay, inside the E. coli, they break it into tiny pieces. As a result, it cannot produce their proteins. Done. That means they used to break the DNA, genetic material of the bacteriophage. Done. So this is nothing but the resistance. So this particular E. coli is exhibiting resistance to the bacteriophage DNA. It is not allowing the bacteriophage DNA or its genetic material to take over this E. coli cell. No, because of the enzymes. Then he extracted these enzymes and finally he found that there were enzymes, they could cut the DNA at specific sequences, and then only it came to know that the enzymes were nothing but restriction enzymes. Okay, restriction enzymes. That means they restrict the growth or development of genetic material inside a host organism. That is restriction enzymes. One important point I would like to add: where exactly you see the restriction enzymes? Only in prokaryotes. In human beings, we don't have restriction enzymes. Just imagine if we have restriction enzymes in our each cell. every day they would have cut the DNA into many pieces in our cell, correct? We wouldn't be surviving. So this is the thing. Okay, so this is the thing. We just write only in prokaryotes it is seen. That means the plasmids also acts like resistance for like different types of chemicals. At the same time, they have restriction enzymes also. They also acts like immune system. They will not allow the entry of other foreign DNA. This is it. Okay, so that is actually the thing. Let me just erase this portion now. Okay. Now we just continue in the restriction enzymes itself. Okay, in the restriction enzymes there are two types. Okay, it was found after 1963. Then because of the research work, the scientists found that there are two types of restriction enzymes, RE. The first type is, let me just write here, methylase restriction enzymes. Methylase enzymes. What is the function of this methylase enzyme? They used to add methyl groups to the DNA. I will just explain it later. Okay, the second type is restriction enzymes. Let me tell you why I have written restriction enzymes again. I am just classifying restriction enzymes into two types. One is methylase, other one is restriction enzymes itself, because in the restriction enzymes you see two types. Because even though they belong to the main class of restriction enzymes, but again I have written the same. Only thing is, in that there are two types. The first one is restriction exonucleases. Other one is restriction endonucleases. Okay, this is the classification system. Again, you get this question for your competitive examination, which type of restriction endonucleases are preferred in genetic engineering? In that restriction endonucleases, you see three types. That is type 1 restriction endonuclease, then again type 2 restriction endonuclease, third one is type 3 restriction endonuclease. In that, in genetic engineering or biotechnology, scientists prefer only type 2. Why? Another question could be added. Why type 2 is used in genetic engineering? Why not type 1 or type 3? The thing is, we need fragments of the DNA, that is gene is required actually. For that we need to allow the DNA with this restriction endonuclease so that they can cleave that DNA into small fragments. But the thing is, we do not want lengthy fragments. We want a short fragments. The fragment of the DNA which is now cut by this endonuclease should be 4 to 8 base pairs. That is, this is the fragment after treating with restriction endonuclease. See, 1, 2, 3, 4, 5, 6, 7, 8, maximum 8. So out of 3, this is the only one which gives that ideal nitrogenous base pair containing DNA after its action. So that's what we prefer type 2. Type 1, it could be more than 20 base pairs. So you are going to get a very lengthy DNA. It's not good for genetic engineering. Type 3 also same. Okay, it could be more or less. So this is ideal, type 2, because it can cut a DNA which contains 4 to 8 base pairs. Please add this point. 4 to 8 base pairs. Add this. Okay, erase it now again. Okay, please write down each and every point. Very important. Now we'll talk about this methyl group or methylase restriction enzymes. Even though it is not necessary for genetic engineering, but let me tell you, in some prokaryotes, because as I said, RE is present or seen only in prokaryotes. Other than that, no. Any other organism will not exhibit or show these restriction enzymes. What I am going to do is, I am just going to draw a DNA. This DNA is, let us just assume that this is viral DNA. So viral DNA is now somehow inserted into the bacteria. So this is the bacteria now. Okay, inserted now into this bacteria, and we know that this genetic material is going to hijack the entire cell, E. coli. Okay, but they have certain systems. As I said, plasmid is also there, and even other restriction enzymes will be there to cut this DNA into tiny pieces and destroy this DNA. It's there, natural system. Along with that, if the two systems failed, okay, The third system is methylase. The methylase is going to modify the genetic material of the virus. Okay, how? By adding methyl groups. So these methylase will add methyl groups of groups to the viral DNA, and if, let us just assume that if introns are present here, don't get confused. Introns and exons are seen only in eukaryotes. Okay, simply for your understanding, I am saying that introns means expressed, sorry, non-expressed. Exons are expressed one. Let us just assume like that for our understanding. If exons are there here, exons means dangerous. If they are expressed, the bacteria cell will die. So they add methyl groups to these exons, that is active segment of this DNA of the virus, and then they just modify the nitrogenous base sequence. Modify, that is interchange the nitrogenous bases. When they can change the change the nitrogenous bases or sequence order, what's going to happen? It gets mutated. If it is mutated, obviously it becomes active or becomes like functionless. That's it, right? So that's it. So it's not going to cause any harm to the bacteria. That is methylase. Okay, don't worry about this. Only worry about restriction endonucleases. Okay. Name the first restriction endonucleases which was isolated from a bacteria for the first time. Okay, we just write that is called as hint 2. This is actually the first ever restriction endonuclease. I hope you can write full. Okay, which was isolated from a bacteria called as, see, capital letter H, that is the genus name of the bacteria, Haemophilus or Philus, small I and small n. So two words I am taking. Influenza, very easy, no confusion here. They have taken from Haemophilus influenza. What about this D? D is the strain name of the Haemophilus influenza. In that particular bacteria, you take any type of bacteria, E. coli. In that also you see around 15 strains, different strains, varieties. So they have taken it from Haemophilus influenza D strain, and the order, extraction order in the sense like serial number, that is number 2, Roman number 2. That is written here, hint 2. It was actually the first restriction enzyme endonuclease which was extracted from Haemophilus influenza by whom? Who extracted this? Scientist name. Please note this. Smith. Next one is Wilcox, and third scientist is Kelly. So three scientists, they actually isolated hint 2 from this particular bacteria. Okay. And let me just tell you one more scientist name called as Nathans. He was another scientist, or like maybe the first scientist to use or show the application of this restriction enzymes in the field of genetics. Note this point, can you write this? So Nathans introduced this restriction enzymes into the field of genetics. So in the field of genetics nowadays, restriction enzymes are used. Application of restriction enzyme could be seen. Why? Because of Nathan, another scientist. Finally, let me tell you, in 1978, after 15 years after discovery of restriction enzyme, Smith, okay, then Nathans, and one more fellow, that is Arber. Arber, Smith, and Nathan. Can you write here? Arber, Smith, and Nathan. These three scientists were awarded Nobel Prize. Why? For the discovery of restriction enzyme, and also for the isolation of the first restriction enzyme, and also introduction of the restriction enzymes to the field of genetics. So ASN 3 were awarded with Nobel Prize. So this is the thing now.